Good morning and good afternoon to all of you for joining our live webinar today, an in silico in vitro pipeline for drug cardiotoxicity screening identifies ionic proarrhythmic mechanisms. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and alongside my colleague, Dr. Alison Obergrusberger, Scientific Communication Manager, will be your moderators for today's events.
Thanks, Jason. So on behalf of everyone at Nanion, we'd like to welcome you all to today's webinar. Thanks very much for joining us. We have two presenters today who are going to tell us a bit more about using automated patch clamp on human-induced pluripotent stem cells. Our first speaker is Dr. András Horváth. András is an application scientist at Nanion. He joined us in 2018 after completing his PhD from the University Medical Center Hamburg-Eppendorf in Germany, together with the University of Szeged in Hungary, where he studied potassium channels in stem cell-derived cardiomyocytes. András will present automating action potential pharmacology in hiPSC cardiomyocytes. And our second speaker will be Alex Clark from Cornell University in New York. Alex did his Bachelor of Science in Bioengineering and Biomedical Engineering at the University of Virginia. And then after spending some time as a research assistant and then a computer science and science teacher, found his way to David Christini's lab in 2018, where he started his PhD. The patch liner was installed in the lab with the dynamic clamp add-on, the Dynamite 8, in June last year. And Alex is going to present some of his work in his talk entitled An in silico in vitro pipeline for drug cardiotoxicity screening identifies ionic proarrhythmia mechanisms. Now I'll hand back to Jason, who's going to just go through a couple of housekeeping rules.
Thanks, Ali. So people are still joining, and just want to make everyone aware of some of the system in the background. So upon completion of the presentation, we're going to hold a live Q&A session with the presenters, and as such, welcome you to submit your questions at any point throughout the course of the presentation. You can log a question in the chat window found on the right-hand side of your screen. Your message will come directly into the queue and be addressed at the conclusion of the last presentation. If you happen to experience any technical difficulties at any time, please inform us via the chat box. Additionally, today's presentation will be made available on demand shortly upon completion of the event, inclusive of a transcribed copy of the Q&A session. And for those of you that want to stay longer, we have allocated an hour for this talk. But we do have the availability to have the presenters stick with us a little bit longer if you have additional questions you'd like to ask. And with that, I will now hand over to András for the start of the presentation.
Thank you so much, Jason and Ali. I would like to also welcome everybody on this webinar and share my screen. Is it good? Everybody can see it?
It's still loading. I'll confirm with you in a second. Yeah, everything looks perfectly fine.
Okay, so yes, again, welcome everybody to today's webinar. And I'm going to talk about applied sodium channel pharmacology on iPSC-derived cardiomyocyte using the patch liner Dynamite. This work was done with a collaboration of Fujifilm and Cellular Dynamics. So they provided us cells for the study. But before, I would like to give a short introduction about iPSC-derived cardiomyocytes. So they are recently a very important model in cardiovascular pharmacology because they have a lot of potential in them. And also they have unlimited access. So as you can see on the schematic figure, you can basically just get a skin biopsy from the patients, reprogram some patient-specific iPSC cells, and either you go in a way to repair the iPSC cells with some molecular biological methods, and then differentiate healthy cells, and then repair the patient's heart, or you can differentiate directly them into cardiomyocytes, which are carrying the mutation, for example, an ion channel mutation, and then you can directly test compounds, blockers, and set up a patient-specific treatment for the patient, or even further study the cardiovascular disease or the ion channel mutation. For this, you need, of course, electrophysiological characterization, such as ion channel measurements, where are still patch clamping is one of the most, or the used gold standard technique. So either you can use manual patch clamping, what you can see on the upper left figure, where a scientist is performing on a system and electrophysiological recordings. And on the right side, you can see that basically using this technique, you approach the cell with a glass pipette, form a strong connection between the cell membrane and the pipette, rupture the cell membrane, and then directly measure the ion channels which are located in the cell membrane. Or you can use automated patch clamping, what you can see on the lower left panel. It's a much more smaller device. And in this case, it's called a planar patch technique, that you use the chip as which is replacing the pipette. And in this case, the machine applies a suction, and then a cell stucks at the chip hole, and then the same procedure forms a strong connection between the cell membrane and the chip. And after rupturing, you can have a direct access to the ion channels located in the cell membrane. Of course, automated patch clamping can be used for iPSC-derived cardiomyocyte studies and could save time and resources for you. So the aim of this study was to apply iPSC-derived cardiomyocytes on action automated patch clamp devices, optimizing recording conditions, and apply pharmacology. And we used our fully automated 8-channel system, the patch liner, combined with a dynamic clamp, which I introduce later. So recording conditions, we used standard solutions for recording solutions. So the internal solution contained higher potassium concentration, of course. We used a sealing solution to enhance the formation of the connection of the seal of the cells and between the cells and the patch clamp chip. And then later we switched on to standard external solution. And to reach whole cell configuration, we used negative pressure in an elevating way until the cell membrane is ruptured, but not more than -290 millibars. The recordings were performed on room temperature, either voltage clamp and current clamp mode, added with dynamic clamping. We used freshly isolated iCaL cardiomyocytes, which were dissociated using Accumox for approximately 40 minutes. You can see on this figure how they looked like after, or the screenshot, you can see how they look like after 40 minutes of dissociation. For the current studies, classical voltage protocols were used, and classical pharmacological application were done. So selective and accepted pharmacological agents. And as I said, we had a special application, dynamic clamping, where we combined an in vitro and in silico technique. So this is the schematic figure of the dynamic clamping. So you can see we have a patch liner, 8-channel patch liner, which is controlled by the patch control software. The patch liner is connected to Heka EPC10 Quattro amplifier, and the amplifiers are connected to the dynamic clamp computer, where we can inject the inward rectifier potassium current, which is many times lower expressed in the cells, or for some reason they're not functional in the cells, and this can cause very technical difficulties to get a reliable resting membrane potential, or like, let's say, measured potential from the cells, because the potassium conductance is rather low, especially when the recording conditions are not optimal. So to overcome this problem, we also used circumvention. So on the right side, on the black trace, you can see an action potential recording where we just used the regular holding current application. And if you see the dark blue trace, we applied 70% circumvention, and on the lower figure marked with B, you can see the injected circumvention. So it's a counteracting membrane potential dependent current, which is not affecting the action potential duration. And on the last figure, you can see the circumvention combined with the inward rectifier, potassium current. And as you can see on the upper panel, that the injected inward rectifier nicely contributes to the late phase of the action potential repolarization. So I would like to go on the results now. So first we did a basic characterization of the cardiomyocytes. On the left panel, you can see the capacitance values, what we measured from the cells. In this case, we were able to record in this study up to 846 cells. This means that every cell which we were able to reach whole cell, we recorded the capacitance value and took into this, this figure. As you can see, there is still a heterogeneity of the cells, and the mean value of the capacitance was around 20 picofarad. On the right panel, you can see an original recording of a sodium channel, and then on the lower, you can see the IV characteristics of the current. And as you can see, in this case, we used 421 cells, which is, I do believe, it's a great and robust number. And then we applied some sodium channel pharmacology using voltage clamp technique. The first one was fleconide, where this is a classical 1C blocker. And as you can see on the upper panels, that the fleconide effectively inhibited the current in a concentration-dependent manner. And also, as an add-on, we tried mexiletine, which is a class 1B, so a strong frequency-dependent blocker. But at a high concentration, it was also able to fully block the sodium current, and the block was also concentration-dependent. And as a special application, we went on with fleconide to try to apply it on the different concentration on an action potential recording. And as you can see on the left figures, the fleconide effectively inhibited the sodium, the action potential amplitude. This is also can be nicely seen on the time course on the lower panel. And of course, it blocked it in a concentration-dependent manner. We also studied late sodium channel activator or potentiator and blockers. We used the CEPA protocol to study the late sodium current. As you can see on the original recording, the 100 nanomolar ATX nicely activated the late sodium current, which could be blocked by ranolazine. And as you can see on the right side, on the bar graphs, we could repeat the experiments, and the difference in the activated current and also the block was significant. We also tried ranolazine on action potential recordings. And as we were also interested because it was shown in the literature that ranolazine has a frequency-dependent effect on the sodium channel. So on the left figure, you can see that in 3 Hertz spacing, the ranolazine started to, with a higher concentration, started to reduce the amplitude of the action potential. And on the right side, we were happy to see that compared to the 5 Hz pacing, which was shown with the black traces, the 3 Hz pacing has a stronger effect, even at lower concentration, which was also significant. So that was a promising result, that we could reproduce literature data that ranolazine has a frequency-dependent block on the action potential amplitude. So in conclusion, the recorded data suggests that automated patch clamp devices can be and are powerful tools to record iCaL cardiomyocytes. Of course, careful development in culturing and dissociation methods are necessary for good success rate. Sodium channel pharmacology could be applied either in voltage clamp, dynamic clamp, and action potential pharmacology on one device using our iCaL cardiomyocytes. Class 1B and C block could be provided. And as a future, of course, we would like to see more class 1A or class 1B sodium channel, more classical sodium channel blockers. And of course, the next step would be apply action potential pharmacology on other cardiac ion channels. So as a last figure, I would like to share that please visit the website of Fujifilm Cellular Dynamics, where we to download the application protocol, where what we made together, how to apply effectively and successfully these cardiomyocytes on the patch liner dynamite. And also you can find information about the patch liner and dynamite on our website. And I would like to thank to Ravi Vaidyanathan, Kara Rieger, Susan De Laura for the nice collaboration and the useful comments what we have during the work. And also I would like to thank Nadine Becker and Elena Dragicevic for their continuous support during this. So with this, I would like to thank your attention, and I would give the word to Alex, and I'm looking forward also to his talk.
Yeah, my name is Alex Clark. I'm in David Christini's lab. I'm entering my 5th year of my PhD, and today I'm going to talk to you about my research titled an in silico in vitro pipeline for drug cardiotoxicity screening identifies ionic proarrhythmia mechanisms. So I have 2 quick slides of background. First, the heart is triggered to beat by an electrical signal that propagates through the cardiac tissue. And this can be measured using an ECG on the surface of the body with the P wave, QRS complex, and the T wave here. Now, the majority, almost all cells in the cardiac tissue are electrically excitable. What that means is they're capable of producing an action potential. And this action potential is generated by the flow of ions through ion channels. Now, if a drug blocks one of these ion channels, such as the IKR channel here, it can lead to action potential prolongation. At the whole heart level, on an ECG, this would look like QT interval prolongation. So prolongation in sort of this T wave and the duration from the start of the Q to the end of the T wave. And this leads to an increased risk of sudden cardiac death. So back in the 1990s, Celdane was a very prominent non-drowsy antihistamine that slightly increased the risk of people developing or developing in sudden cardiac death. So as a result of this and a few other prominent examples, regulatory agencies developed the ICH-S7B guidelines. And these guidelines effectively eliminated the approval of cardiotoxic drugs, but this was at the expense of specificity. So these guidelines included animal long QT studies and studies primarily on this IKR channel. Now, as I've mentioned, these were not the most specific, although highly sensitive. And as a result, potentially beneficial drugs were and are being labeled as cardiotoxic. So a big part of the field, and András gave a great introduction to the use of human derived cardiomyocytes, a huge part of the field is moving towards using these iPSC-CMs so that we can study drugs on human tissue and study multiple ion channels at one time. So András gave the example of sodium, but there also is interest in a few of these other channels as well. So today, what I'm going to talk to you about is first, how we optimized a voltage clamp protocol that can estimate ionic current density in a heterogeneous population of iPSC-CMs in our lab. Then I'm going to talk to you about how we acquired some experimental data, both action potential and voltage clamp data, use this to identify surrogate markers of cardiotoxicity and ion channel targets. And then finally, some very new data that we've just collected in the last 2 months that essentially extends the methodology in step 2 here to a high throughput system. And András gave a really great background on the Nanion patch liner. So throughout this talk, we use the average Kernick-Clancy iPSC-CM model. And here I'm showing, if I just applied a 50% IKR block for the Kernick-Clancy model, you can see this nice action potential prolongation that would be characteristic of such IKR block. Now, the cells that we use in our lab, which we purchased from Joseph Wu at Stanford University, under the experimental conditions that we are using, we do have cells that have spontaneous behavior, but we also have a very heterogeneous population of cells. And some cells, due to either seal resistance or just the ionic current distribution or densities of these cells, we have resting membrane potentials that are quite high and sort of unable to generate an action potential here. So in our lab, we use a method called IK1 dynamic clamp. You got some background in the previous talk. What this does is this in real time measures the voltage in the cell and then calculates an injected synthetic current that will then hyperpolarize these cells so that we can generate action potentials at 1 Hertz. And this IK1 dynamic clamp was implemented in our lab by Drew Tilley, who's a postdoc. I'm going to outline just how we planned these experiments out. So first we start, this is all manual patch clamp at this point. First, we start with just getting electrical access to an iPSC-CM. We acquire action potential data both before and after drug application. And we can use this data to identify surrogate markers of cardiotoxicity, so action potential prolongation. And then the question is, how do we identify the affected ion channels from the same cell? So how do we get at the mechanism that may be causing this? And in this study, we've designed or developed a method to design voltage clamp protocols that can identify these ion channels. So we can say, okay, we see quinidine prolongs this action potential. And with the voltage clamp protocol, we can say it is because quinidine is blocking both IKR and ITO. To do this, we used a computational optimization method to automate the design of voltage clamp protocols that can identify ion channel targets during iPSC-CM drug studies. So we use the Kernick-Clancy model, and we use this fitness function that I'll talk about in just a minute. And as an example, over here on the right side, we're developing voltage clamp protocols. So voltage clamp, you're setting voltage, clamping voltage here, and then you're measuring the current passing through. Or in these computational experiments, just measuring the current that's generated in this Kernick-Clancy model. For this example, I'm going to talk to you about a protocol that is especially good at isolating the L-type calcium current. Yeah, so we're measuring current. Now, if we look at this fitness function, the way that we've designed the fitness function is we take the absolute value of L-type calcium at every single time point, and we divide it by the sum of the absolute value of all other currents. And that will generate this percent contribution plot here. You can see as we step from -80 up to -40, the Kernick-Clancy model generates 85% of the total membrane current is actually from L-type calcium. So how does this help us in a drug study? You can imagine if we applied a drug that blocked L-type calcium, something like verapamil, you may see a shift in this total current. Now, if we zoom in just on this region, because the Kernick-Clancy model predicts 85% of the current here is L-type calcium, we could say if we see a change in this region in an experiment, this change is likely due to L-type calcium. Now, one thing that I really want to emphasize at this point is that not all cells will have a lot of L-type calcium. Perhaps in some cells, this region will only be 20% L-type calcium, or in some cells, it'll be 95% L-type calcium. That's sort of the nature of using such a heterogeneous population of cells. But the important thing is that no matter what cell you're using, L-type calcium will likely be present here. And so a shift in this region would be indicative of some sort of L-type calcium block, or likely indicative of some sort of L-type calcium block. Okay, so we use this genetic algorithm for 7 different currents here. And for, because we're looking only at the maximum, so that 85% on the previous protocol I showed you, there's only a small region of these protocols that we're really interested in. And so what we can actually do is we can strip away the majority of these protocols, which we do here. And then we put them all together with a negative 80 millivolt holding step between each. Now, this is the protocol that we then use to identify the ion channel targets. All right, so I'm going to jump into the experimental results now. I'm going to start with the action potential data. This is showing a cell that was treated with cisapride. So cisapride is a strong IKR blocker. And this is showing sort of the characteristic action potential prolongation that I've already shown a couple of times. When we look at the small population study that we did, we actually did not see a significant difference between a DMSO control group here and our cisapride treated group here. I'll come back to speculation as to why this may be in just a minute. But in addition to cisapride, we did these same experiments with verapamil, quinidine, and quinine. And quinidine and quinine, being strong, more general potassium channel blockers, they are known to prolong the action potential. We saw action potential prolongation, significant action potential prolongation, whereas verapamil, it's a stronger L-type calcium blocker, blocks a little bit of IKR. We did not see significant prolongation here. Now, this sort of begs the question, why do we see prolongation for quinidine and quinine, but not cisapride? And there are a few hypotheses. One hypothesis could be that IKR is not present in these cells. An alternative hypothesis is perhaps that IKR is just not the dominant repolarizing current in these cells. So this is what we really designed the voltage clamp protocol for, to help us answer that question. So which ion channels are blocked to cause these action potential duration changes? Or in the case of cisapride, no changes. So now I'm gonna just zoom into the IKR experimental results. So this is showing one example cell that was treated with quinine. So quinine is a strong IKR blocker. And I'm gonna just zoom in on this IKR segment of the protocol. If you look here, there's a reduction in total current, a negative shift in total current, which would be characteristic of such IKR block. And when I compare the change in the IKR segment here of the control group to cisapride, verapamil, quinidine, and quinine, you can see significant reductions in cisapride, quinidine, and quinine. And so we felt pretty confident at this point that IKR is indeed present in these cells for the most part. However, perhaps, like I said on the previous slide, it may not be the dominant repolarizing potassium current. In addition to this, we also did a functional t-test. In a functional t-test, we find the significance value comparing an average change in our DMSO group to the average change in our cisapride group at every single time point. And this panel up here is showing where there is a significant difference throughout the entire protocol. So for cisapride, we see a really nice significant difference in this region. See the same thing for quinidine and quinine. We didn't see this for verapamil. Verapamil does block IKR at this concentration, about 20%, but we were really only able to pick up these strong blocks for these drugs. Now I'm going to summarize some of these findings and discuss the findings for the other ionic currents. So as I already mentioned, we did find significant prolongation for both quinidine and quinine, and not for cisapride or verapamil. These two drugs are known to be or are labeled as proarrhythmic. Cisapride is also labeled as proarrhythmic. So we were a little disappointed to see that, but it was nice that we did identify IKR block here. Now, these percentages I'm showing here, these are not percentages that we generated. These are percentages that came from this 2016 Crum study. So you would expect a very strong block of IKR at this concentration. For verapamil, we did detect the block of L-type calcium. For quinidine, we detected a block of IKR and ITO. And then finally for quinine, we detected a block of IKR. Now, it's important to note here that we detected all currents that this Crum paper estimates to have been blocked by more than 30% at the concentrations used. And we did not have any false positives. In addition to these findings here, we also found a previously unreported block of funny current by quinine. So we were interested in this. We wanted to look into it further and see if we see what this sort of looked like on a individual cell level. If I zoom in on this funny current region of the voltage clamp protocol, you can see there's a dip down to negative 120 millivolts. This is very characteristic of how you would activate funny current. You have to be holding at a very large negative value for quite a while. And you see from pre to post drug application, there is a positive shift in total current. And this is sort of agrees with the idea that you'd be blocking funny current because funny current is an inward current at large negative voltages. Now, this is just one individual cell. This figure is a little complicated, so I'll try to walk through it a bit slow. Up here on the top panel, I'm showing the significance where there's a significant difference between the change in quinine and the change in the DMSO control groups. You can see throughout this region, there is a significant difference between these groups. Now, at the bottom here, I've plotted the Kernick-Clancy funny current. And you can see throughout this region where there's this significant difference between these two groups, this Kernick-Clancy funny current is turned on. So this looked really nice. And what was nice about this is it wasn't just when funny current was expected to be an inward current, but also when you step up to positive 15 millivolts and there's a reversal of the current there. So we were pretty convinced at this point that quinine did indeed block funny current, but just to be completely sure, we did a dose response. We conducted dose response studies using an expression line cell and the Nanion patch liner here, and constructed this dose response curve. And at the concentration used in this study, we would have expected 32% of funny current to be blocked by quinine. Now, just to summarize this portion of the talk, the first thing that we did here is we optimized a voltage clamp protocol, and then we collected action potential, IK1 dynamically clamped action potential data before and after drug application, and voltage clamp data before and after drug application. We use the action potential data to identify surrogate markers of cardiotoxicity, such as APD90 prolongation, and then use the voltage clamp data to determine ion channel targets. So, quinine block of funny current. And then finally, we confirmed this finding here by constructing this dose response curve and using expression line cells. So this paper was just accepted in the last couple of weeks. So yeah, it should be up online now if you're interested in reading more about this. Now, one quick thing that I wanted to highlight here is that despite the heterogeneity of the cells that we used in this study, the model, which looks quite different from the cells that we used in this study, still generated a protocol that worked, right? And so this is a subtle departure from how this model is often used, which is just typically to simulate experiments that are either expensive or sort of not feasible. So instead, we use this model to actually design the experiments. So we saw this model as not just being valuable in generating this voltage clamp protocol, but we also saw this study as being a really nice validation of the model itself. This model is able to generate something that sort of seems to agree with the kinetics and the dynamics of these cells up here. It's really nice. Now, with all that said, we were really happy with that study, but that approach is slow and only collects data at one concentration. And so we asked the question, can we increase the throughput and collect data at multiple concentrations by using an automated patch clamp system? Fortunately, I don't have to go into a whole lot of detail about this, the patch liner. So I'm just gonna jump into how we've designed these experiments to extend the manual patch clamp studies that I just went through. We have the 4-channel Nanion patch liner, and what we aim to do is collect data at baseline, then 3, 10, and 20 times the effective free plasma concentration. And the way that we've designed this protocol is we start by collecting voltage clamp data. Now, this voltage clamp protocol looks a little bit different. We use the same optimization approach, but we just added a late sodium component to the model as well. And so we start by collecting voltage clamp data. switch to action or current clamp mode, and we collect IK1 dynamically clamped action potential data. And while we're acquiring that data, we will apply a drug, continue to record action potentials, and we can compare that change here, switch back to voltage clamp mode, acquire that data at 3 times the effective free plasma concentration. And we do that switching between voltage clamp and current clamp mode at 10 and 20x the effective free plasma concentration as well. And then finally, we do a wash step. Now, this work was done. We've been working on getting this protocol working for probably 6 to 12 months now. And I was working in collaboration or in partnership with Siu Wei, who's a researcher in our lab. She's a very talented experimentalist. So I'm able to talk here, but we really did all of this work in unison. Okay, I figured because I'm here and this is somewhat of a work in progress, and you all would like to see the results from the patch liner. This data is very preliminary. But I think that it really shows you our intentions and where we're headed with this. So this is showing a example cell that was treated with moxifloxacin, which is an IKR and IKS blocker. I'm showing changes first from baseline to 3x, then 3x to 10x, 10x to 20x, and then 20x to DMSO. What was nice to see is that for this one example, we see prolongation from 3 to 10x here, from 10 to 20x, and then a nice sort of shortening after this DMSO wash. Now, if I look at the percent change in APD90 for this tiny sample size of N=3, as I said, very preliminary, we're collecting more of this in the coming weeks. If you look at APD90 change here, first base to 3, 3 to 10, 10 to 20, and 20 to wash, we're really hoping that this trend holds. In particular, seeing that all 3 of the cells that we used here saw prolongation from 10 to 20, and then shortening from 20 to wash. We've done these same studies, and we are continuing to do these studies with verapamil, flecainide, and chloroquine as well. And the purpose of doing this is because between all of these drugs, we're actually capturing all of the CIPA suggested channels. But what is the mechanism? So I'm gonna just show you one example again for moxifloxacin, and we're gonna zoom in right on this IKS segment. So as you can see, this is a long holding step close to positive 50 millivolts here. IKS should be gradually turning on over the time course of this portion of the protocol. Now here, the black curve is showing the baseline. And as the curves get darker and darker, those are increasing concentrations of drug. You can see that for all 3 of these cells, it goes from black here to this dark red, indicating an IKS block. So a reduction in total current. IKS is an outward current here. This means that it could be IKS block that's causing this. And then after a wash, you see this gray curve has a nice recovery. Similar to these same 3 cells that I had shown previously, we're doing the change in membrane current from baseline. So from at baseline, it's baseline minus baseline here. So this should be 0 for all 3 cells. Then at 3x, 10x, and 20x, we're seeing reductions at all 3 of these concentrations. And then at a wash, we see some amount of recovery, which was nice to see. So we're really hoping to see this trend hold as well. And we're doing the same thing for these other drugs in this study. So to summarize, I started by talking about how we optimized a voltage clamp protocol that can estimate ionic current densities in our heterogeneous population of iPSCs. How we, with using manual patch clamp, acquired action potential and voltage clamp data before and after drug application. Use that to identify surrogate markers of cardiotoxicity and ion channel targets. And then finally, how we're attempting to extend this approach from step 2 to our automated patch clamp system so we can acquire more data faster and at multiple concentrations. And with that, I would like to acknowledge my lab, in particular Drew Tilley, who implemented the IK1 dynamic clamp, Siu Wei, who helped with all of the development of the protocol, or sorry, the automated patch clamp protocol, Trina Krogh-Madsen, who is really is one of my advisors, and it's a really valuable help when it came to the modeling portion of things. And then Nadine Becker, who sat through a lot of be asking quite a few questions as I tried to get the protocol working on our Nanion patch liner. And finally, if you want to reach out to me, this is my email address, and I look forward to your questions. So, thank you.
Thanks very much, Alex and András. Very nice talks. We have had a few questions come in. So if András is also online, I think he is, then I'll start with some questions that came in during his talk.
I'm here.
Someone asks, what are the RS values in your experiments? Because good RS is critical for sodium IV curves.
Yes, that's true. The RS values were rather low, so most of the times were below 10 mega ohms. But I don't know by heart. I need to check this.
I had a question also. You showed the IC50 for flecainide on action potentials. Do you know how that compares with the IC50 in voltage clamp mode on NAV 1.5?
Again?
You showed the IC50 for flecainide on the action potentials. Do you know what the IC50 is in voltage clamp mode on NAV 1.5 and how that compares with what you saw on the action potentials?
Yeah, so during the voltage clamp experiments, the IC50 was around 10 micromole. Mm-hmm. And in the action potential, it was a little bit higher, so it was around 24. I don't know the reason why it's so different at the moment, or there is a slight difference, so 2 times. But yeah, it's a good question.
Was there a lot of variability, or were they quite similar, between cells?
Variability, yeah, in voltage clamp there were more variability, but also we have less N numbers, like compared to action potentials. Some cells were more sensitive also, so there was a bit of variability. So probably would be nice to increase the N numbers, especially in action potentials. They were a bit more variable.
And then another question that came in for you, András, as well. What extent does the 110 mm potassium fluoride affect the sodium current pharmacology?
As far as I know, it should not affect the sodium channel pharmacology. I mean, as far as I don't really know any affecting property of fluoride on sodium channel.
Okay, then another subsequent question. Is it correct that the APD90 is about 70 ms in the iCaL cardiomyocytes?
"70 milliseconds. The APD90 varied. It had also a bit of heterogeneity, since we have freshly isolated cells. Depended also how strong was the calcium current, calcium channel expression, or the calcium current density on or calcium current amplitude on the in the cells. So there were shorter action potentials and longer, but generally the duration of the action potentials, so the mean actual APD90 values were over 100 milliseconds, 100, 120.
And then another question that came in is, does this work only with these kind of cells, or does it also work with cells from patients expressing mutant channels? And is there any data for that?
So certainly it works on other cells as well. And so we made most of the studies with iCaL cardiomyocytes, but certainly we tried different different sources, and they can also work nicely. But you have to keep in mind that this particular dissociation technique and application, so mainly focusing on dissociation, works on the culturing condition, but what we followed based on the guidelines of FCDI. Of course, if you have a different culturing or coating material, can be that you need to optimize your dissociation according. So, for example, matrigea can take longer time. If the cells are growing on matrigea, can take longer time. Some of the users use different enzymes, and they work nicely for them. In this particular cell, this worked the best. So, but certainly, of course, you can use any sources of IP.
You could also introduce some kind of mutation, right?
Yes.
Into the cardiomyocytes, and they would work. I think there are some
absolutely.
other people who have done that, who have published on that.
Yes.
And then a question actually for both of you, maybe you can each comment on this. The models consider 37 degrees conditions Celsius. Are your experiments also at 37 Celsius, or how do you adjust it?
My experimental conditions were room temperature mainly. So
our manual patch clamp experiments are 35 to 37, and then for the iPSC experiments, we've started to use set 35 degrees as well. On the patch liner.
Also on the patch liner.
Yeah, sorry about that.
Yeah, so you're using the temperature control of the patch liner and set that to 35 degrees. Another question that came in is, this is for you, Alex. What is the advantage of a single voltage clamp protocol for all channels over multiple individual protocols for each channel? Wouldn't a P/N leak subtraction work better with individual protocols?
So yes, so if I'm understanding the question, the application here, we didn't really take advantage of relating the individual voltage clamp response to the action potential from that same cell. So we see the value of this being that we're able to acquire a lot of very dynamic data about individual ionic currents, each of the currents, very quickly, and hopefully we could relate that data down the road to the shape of the action potential. Now, I do think that if we were able to have a much longer sort of protocol, and we were really just focused on acquiring data maybe after one additional concentration, then perhaps that may be a better approach. But I think that we sort of saw this protocol as being twofold, both for being able to detect drug effects in a population of cells, but also being able to relate these the dynamics of the current to the action potential itself for an individual cell.
Maybe this is a bit of a follow-on question from that actually then as well. How about using an action potential waveform as a stimulus in voltage clamp?
Yeah, I think that would be really great. There's the classic sequential dissection approach, which is really quite nice. We're a little limited by our use with Patchmaster at the moment. I'm not familiar with how to use a custom waveform. I know how to use ramps and steps, but our lab has in the past used action potential clamp, and even an action potential clamp for the given cell that we've been recording from. So I just think that would be just like add additional very rich data set for us to consider.
can I have also a question to you, Alex? Not really a question. When you show the cisapride data, that somewhat it wasn't so effective on the action potential duration, right?
Yes. Mm-hmm.
Yeah. Just a comment. We also worked former times in Hungary with cisapride, and we also did in vivo experiments. And compared to dofetilide, which is like a very classical ICaL blocker, cisapride was, and so for example, the animals were much less sensitive to cisapride. And then we saw actually on the voltage clamp data, so the patch clamp data, that it's nicely blocking the IKR. We were also a bit surprised that somewhat cisapride is not that aggressive somehow than other IKR blockers. That was also very interesting for us. And
it's interesting you saw the, oh, sorry.
Yeah. So for example, these were rabbits, what we used in the in vivo studies, and as far as I remember, never saw, let's say, also an arrhythmic event, which in case of dofetilide was much more frequent, that arrhythmic events appeared during the experiments. And did you see maybe that, I mean, with quinidine or quinine, that the cardiomyocytes, the iPSC cardiomyocytes were more sensitive than, let's say, a regular, like an adult cardiomyocyte? Because this is what we saw in Hamburg, for example, that the iPSC cardiomyocytes reacted much more stronger to IKR block, for example, than to let's say a regular cell, like an adult cardiomyocyte.
They reacted more strongly to an IKR block, or because you said
iPSC cardiomyocyte. Yeah.
Oh. Yeah, I mean, cisapride is the only data that we really had. I mean, we're now collecting flecainide data as well, but not to get too off track, the sort of expression of IKR in cells when doing automated patch clamp is quite different from when they're when we use the manual patch clamp. So yeah, I didn't notice, we didn't do any comparisons to adults. But I was just thinking, I just figured, like I said, sort of during the talk, that because quinidine and quinine seem to be more general potassium channel blockers, I mean, quinine blocks sort of every potassium channel, it seems. That's why we were seeing that prolongation more consistently. It was very sort of, those drugs are robust to producing prolongation, whereas cisapride, being so specific, just didn't see it. Yeah. Good question though. Yeah.
We had another question that came in, which is, why did you select the Kernick-Clancy model? Did you try Pussy's model?
Yeah, so we use the Pachi model as well. And we actually use the Pachi model as more of a validation. So there are more details of that in the paper. We selected the Kernick-Clancy model because it's really an average of data from numerous labs, and it was trained. A lot of the training data that went into the Kernick-Clancy model came from Joseph Wu's lab, which is those are the cells that we get as well. So we just kind of thought that it would be a bit closer to the cells that we were using. And when we did look at whether the voltage clamp protocol isolated currents from the Pachi model during the same times, it did, which was a really nice validation, because we were able to see that, okay, assuming that there is a large amount of heterogeneity in the Pachi model, represents one example of a different cell. This protocol was still able to isolate current with that model.
Going back to the temperature question that we had earlier on, somebody asked, did the cell seal at 35 degrees C? Because that's impressive, because it's even tricky in manual patch clamp.
So what's nice about the temperature control is that you can seal at a lower temperature. So you can seal at 25 and then increase the temperature up to 35. And when we got this patch liner, it was after doing a year of manual patch clamp, it was pretty amazing. So yeah, I had a lot of trouble getting seals at 35 to 37 with manual patch clamp, but now with the patch liner being able to seal first at 24 and then increase the temperature, to 35 is quite nice.
Okay, so that's your sort of experimental protocol that you seal at room temperature and then raise the temperature. How long does it take then to get to 35 degrees approximately?
And I wouldn't say that this is my protocol. This is suggestions from people who have a lot of experience at Nanion. But it takes under 5 minutes, 3 to 5 minutes perhaps, yeah.
I used the same approach when I tried temperature controlled experiments, what you mentioned, Alex. So yeah, it takes a couple of minutes, but also you can implement it that it's automatically switching on at one point and switching off when you start a new experiment. So I think it's pretty convenient and makes it more, it helps to perform a good experimental condition.
Mm-hmm.
And a follow-up question for you, Alex. Is current rundown a problem in your experimental conditions?
Yeah, that's a really great question. So in the manual patch clamp, we were using perforated. And with the perforated patch clamp, we didn't observe a huge amount of rundown. We didn't explicitly do any sort of calcium test to check for that. But it was something that we were really wary about and concerned about when switching over to the patch liner, because with the patch liner, it's kind of difficult to do perforated patch. Now, the internal solution has EGTA in it, which chelates the calcium. So from what I've seen thus far, I actually do think that 15, 20 minutes in, the action potentials look pretty similar. But I think that we would have to do a bit more directed investigation for me to answer that question with more confidence.
Perfect. Thanks so much.
Have
you tried to do perforated patch on the patch liner?
Yeah, we did. We had reasonable success. It's funny because so with the manual patch clamp, we front load the tip of the pipette with a solution that doesn't have the perforating agent, and then backfill it with, we were using amphotericin. With the patch liner, the thing is that we were using beta-essin. We found that worked a little bit better. Because it's in direct contact with the cell, we would basically get access within seconds of seal. And it was hard for us to really tell if we were getting access from the perforation of the perforating agent, or if it was from rupture. So we decided, all right, let's just try to get this protocol working for with rupture, and maybe we'll go back to perforating using a perforating agent if we run into issues down the road. So we may reinvestigate that, I guess. Yeah.
So we're just about at the hour mark. I think that's a good transition for today's webinar. András, Alex, thank you so much for your great presentations today. For everyone in attendance, we will make this available on demand. So in about 2 days' time, you should receive an email with a copy of this, also a link to all the Q&A that we have. And if you have any additional questions after rewatching, I'm sure that Alex and András will be more than happy to answer any of your questions. And with that, I'd like to thank you one more time, and we'll see you at our next webinar.
Thank you so much.
Thank you.
Thank you.